Well, thank you very much. Hopefully, I don't have to repeat an awful lot of the introduction after Dr. Wedgwood's talks, because we are working in a similar area. I am actually doing L-arginine supplementation, but my study is in adult severe asthmatics, and you'll be seeing a little bit of that data. So I'll talk just briefly in the background about a couple of things. One, why nutritional supplements are of huge interest right now in severe asthma, and a little bit about the rationale for our clinical trial. Then I'll be talking about the advantages I've seen with the Metabolomics approach over the past year, really, and how it's validated our mouse-to-human approach in our work, and how it's informing future studies. So asthma has become extraordinarily expensive really over the past 10 years. It's got an awful lot of press, as probably many of you have seen. If you look at the bottom of that table right there, you can see that the asthma drug market is expected to increase from just $15 to $23 billion. Most of that is from drug costs, as you can see on that right figure over there. And that's because of the coming biologics. Anti-IL-5, for example, is going to be approved in the next month. And there's anti-IL-13, anti-IL-4. It's following a lot of the oncology market with monoclonal antibodies. So there's a lot of interest in nutritional supplements, and I've done some work with the Metabolomics core in the past on omega-3 fatty acid supplementation. Just to give you an idea, there have been 20 studies of enriched fish oils in severe asthma over the past 15 years. And you probably know the vitamin D story. Right now, on clinicaltrials.gov, there are 35 clinical trials of vitamin D in asthma going on right now. All of these that I've listed right here are of interest clinically in being studied in asthma. The figure on the right, just for example, is a pilgrimage in India where they treat kids with live sardines, and they've been doing that for over 150 years. So while we study it, this is well past the clinical trial phase in some areas of the world. 43,000 fish were given to kids a couple of years ago. So L-arginine, and you heard Steve Wedgwood talk about that. He's interested in a supplementation study in pulmonary hypertension. It's been looked at mostly in vascular disease. If you look at some of the clinical studies there have been many folks looking at L-arginine supplementation at huge doses, IV, MPO, and it has a variety of effects. Many patients are actually already taking this, as they are with other amino acids as well. It comes in a variety of formats, just simple L-arginine from GNC, but also in some fairly fancy names, such as you can see on the bottom, NO enhancer. The most interesting study funded by the NIH in the past 5 years really has been sickle cell disease, looking at vascular disease, both as a bronchodilator and a vascular dilator in acute chest crisis. So there are a couple of pilot studies in L-arginine, and this sort of validates the approach of L-arginine deprivation. So there's one study 10 years ago now looking at plasma arginine levels in kids and adults who came into the emergency room, and you can see plasma arginine levels are about half what the control levels were at the time of admission to the hospital, and serum arginase activity measured by urea formation about twice as high, and that reverses as soon as asthma gets better, per se. So we were interested in doing this in adult severe asthmatics. We did also a pilot trial a few years ago now, knowing that arginine levels are depleted at the time of peak inflammation. So we were working on the dose, and we found that this dose of 0.05 grams per kilogram of ideal body weight twice daily increased arginine concentrations in plasma significantly over 3 months. We're also interested in following this biomarker on the right, asymmetric dimethylarginine, which is a nitric oxide synthase inhibitor, and has been shown to be of some concern in the cardiovascular studies in the past. This was done in just 20 studies, but it was the patient population that we are using now in our clinical trial. So really the paradigm in asthma is this, and also in other diseases, that the arginase enzymes and the nitric oxide synthase enzymes are in competition for the substrate L-arginine. Sometimes during peak inflammation there may not be enough L-arginine to produce nitric oxide. Instead, you have high levels of arginase, which I'll show you, which leads to smooth muscle. It leads to polyamines that have effect on smooth muscle. Also high levels of proline, which is the building block of hydroxyproline, which we think is part of the remodeling response in asthma and severe asthma in particular. And one thing Steve didn't mention, if you look at the lower figure here, there's an issue of uncoupling of the nitric oxide synthase enzymes, which may be part of the issue with arginine deprivation. With low levels of tetrahydrobiopterin or L-arginine per se, you may not actually get citrulline formation through the nitric oxide synthase enzymes. Instead, you may end up with superoxide and peroxynitrate formation, which is extremely problematic, obviously, in airway epithelial cells. So we approached the West Coast Metabolomics Center and said, how do we get a better handle on all of these amino acids and small molecules downstream of the arginase and NOS enzymes? The hypothesis of the large clinical trial is supplementation with arginine will decrease lung inflammation and improve asthma outcomes. For this pilot project, we felt that arginine metabolites downstream of arginase would correlate with increased allergic inflammation in mice and also asthma outcomes in humans, and that the relative concentrations of these metabolites will really highlight what's actually happening to L-arginine metabolism. So the center, along with us, decided to take a staged approach, which has been terrific. First, they wanted to validate a new method for doing this, HILIC, hydrophilic interaction, with the underivatized samples. This required some method development, which I'll show next. Then we went on to use our mouse samples and then the ongoing human trials. So there's a couple of things that were of interest to us. One was to measure asymmetric dimethyl arginine. If you see that pink peak halfway down. In some of the other mass spec approaches, apparently it's hard to separate ADMA and SDMA. ADMA, as I mentioned, is a nitric oxide synthase inhibitor, which was of specific concern. They were able to do this method development and separate those two, which very nicely. And the second thing they did with the method is we have now, with some chemists and some engineers, labeled arginine with a PET avid fluorine labeler so that we can actually determine non-invasively by PET where the activity of arginine and later arginase is, and they've been able to separate that for us. So it's already led to some new projects for us and new grant formations. So our mouse model is this, that this is just a figure of us using a pegylated formation of arginine that we made. We normally sensitize in mice and expose mice to ovalbumin or to filtered air over basically a 2.5 week period after the sensitization, and then we sacrifice and collect and measure lung function on these mice. And if just as up on the upper right there, this is arginase 1 activity, and that's a Western blot up above. Normally you don't see arginase 1 in the lung, but it's highly induced by IL-13 and other Th2 cytokines upon exposure to ovalbumin over time. And down below it shows that we can actually manipulate arginine levels. That's arginine on the y-axis there, and on the bar on the far right is giving an arginase inhibitor. So you can see giving an arginase inhibitor, we're able to actually affect arginine levels in these mice. But we wanted to know what actually happens to the concentrations of many of these amino acids. So when we see a time of peak inflammation, which is 2 weeks of exposure for us, and you can see that the lung lavage cell count there is about 2 million in the ovalbumin. The filtered air is essentially about 20, 30,000 per se. You can see that arginine is depleted. So this sort of validates the paradigm per se in these mice. You do see higher levels of asymmetric dimethylarginine, which is what we expected, as well as downstream products of arginase, such as proline, which is an adverse outcome. They weren't statistically significant in this small n size, but I expect it's going to hold up. So that's our mouse model work that we've done for some time. We went on to this human clinical trial with the Metabolomics Center. This is an active trial. It's about 80% completed, but it's still going on. These are adult severe asthmatics. I won't go through all the inclusion criteria, but just to say it's extensive. And they're grouped by a biomarker, which we use clinically, nitric oxide, exhaled nitric oxide. And the hypothesis is that really the subjects with the low exhaled nitric oxide at baseline, less than 20 parts per billion, are really going to benefit more with the arginine supplementation. It's a crossover design trial, as you can see there, where they get arginine and placebo for 3 months each. We've now been able to add the metabolites as a secondary endpoint. And just to show you where we are preliminarily so far, these are obviously blinded samples still. I went to the Metabolomics Center. So we're interested to see if we see any correlation with lung function and symptoms. So that on the y-axis there is FEV1 FVC, which is a measure of airflow obstruction. So the lower number suggests worse lung function per se. So as you can see, there's no significant correlation with arginine, increasing arginine levels, which is what I suspected, but I did not expect what we found on the right. Creatine, which is the most common amino acid supplement taken by many, many people, many patients, was increasing levels of creatine correlated very strongly with worsening lung function over time. It's interesting because creatine is actually used in some clinical studies in lung disease as well, particularly COPD. So this needs to be further followed by us. Asthma control test scores also we were able to correlate with these metabolites of arginine. No significant change overall with increasing arginine levels, but you can see they're highly grouped at low levels. But also asymmetric dimethylarginine and creatine, increasing levels of them correlated with worsening asthma symptom scores. I expected it with ADMA, not so with creatine, which is very commonly taken. And obviously we're interested in this ADMA to see how good the correlation is with increasing levels of glutamine and ornithine. Glutamine is also interesting because a lot of asthmatics take L-glutamine and have found some improvement in outcomes, including some of our patients. So this has given us some feedback already, even though the study's not done, of where we're going to go next. For example, citrulline and other amino acid supplementation. As you can see, and this is lung tissue citrulline, lung tissue percent change with citrulline and arginine for both, and also in plasma for both citrulline, ornithine, and arginine. You can see that perhaps with citrulline, we may be getting better levels in the plasma, maybe not so much in the lung, and this is going to be a future direction for us doing citrulline supplementation. So in conclusion, so far, we've seen that creatine and ADMA correlate with worse lung function and asthma scores. We expected one, but certainly not the other. I think L-citrulline may be a direction that we're going next in terms of supplementation. Others are interested in this as well. We've also found, thanks to them, we are able to label L-arginine and arginase inhibitors with PET avid tracers, which is going to be important for looking for the site of activation. And it's already led to several proposals for us as well, including the one on the lower right, which is a response, an RFA, looking for non-invasive biomarkers for pediatric asthma. We've developed a phone-based spirometer that we can put some of these breath metabolites in. All right. And overall, this L-arginine is part of a paradigm for us for treating non-allergic obese asthmatics with statins, which we're looking at arginine, fish oils. It looks quite different than what's been done in the past. The two most important people here on this list are Jennifer Pratt, a postdoc in my lab, and also Megan Showalter, who's a fantastic graduate student in Oliver's lab. Thank you.
Thank you. Questions? Yes.
Very nice talk, Nick. So question I have is basically you alluded to the polyamine biosynthesis pathway, putrescine, spermidine. Have you noticed any impacts on that pathway as you're seeing these changes in arginine and ADMA?
No, the ones that I've picked out so far, John, are the ones that have been statistically significant based. We have about 25 or 30 of these that we've done analysis for, but these are the ones that stick out so far. Yeah.
So you do have the measures of the polyamine biosynthesis in there.
Yeah, I'm not sure we have all of them, but we are able to look at spermidine. Yeah.
We have time for one last question.
Thank you very much. I didn't follow up literature, but maybe you can give me an answer. Differences in citrulline uptake by liver. If you provide the arginine by the oral route, you get a considerable amount of citrulline appearing there, but there seems to be a difference between rodents and humans.
Right, and we have not gotten to that very well with our mouse studies right now. So we've actually, the pharma companies are always asking me if I'm making inhalers of these to see how they're taking up, whether the airway epithelium. Patients don't want to take these large supplement pills, and that's been the biggest drawback in my study per se. But when we come to the mouse models, we've done everything IP, IT, and IV. So you're right, we have not actually studied to see what these concentrations are going to be like in mice by giving it by gavage, which I think is going to be extremely important to get to the point of what the uptake is. But I do think actually it's because of these pills being so large, I think it's going to have to be an inhaler that eventually gets developed as a therapeutic.
Thank you. Let's have another round of applause for our 6 fantastic speakers.